videos now. That makes me super happy, and I wanted to put another one together for you guys. So this one's gonna have some similar information, fairly basic information about what DNA is, but kind of focusing on the genetic basis of diseases. So I get a lot of questions in my genetics videos along the lines of like, do you think mental health disorders are genetic? Things like that. So I'll kind of touch on that and touch on the genetic basis of disease in addition to what DNA is and how it makes us who we are. So I hope you enjoy. I also recently found out that some professors and genetic counselors at my school, where I'm in genetic counseling school, found my videos and watched them. So that's extremely embarrassing, but if that's you, then hello, thank you for being here. It makes me nervous finding out that like geneticists at my school watch my videos, because it makes me scared I'm gonna say something wrong. So if I say something wrong, I'm sorry. Okay, let's get started. So we are gonna start talking about something that I haven't really talked about on here before. Not really my forte, but I kind of looked up stuff for this video. So we're gonna talk about what makes up DNA. And DNA, as I've talked about before, is essentially the instruction manual for everything going on in our body. So I kind of think of it like a recipe book for your body that tells you how to make all of the proteins that your body needs, like the ones that make your heart pump, the ones that make your liver function, the ones that make your skin, all of the proteins. The DNA is the recipe book for that. And the most basic building block of DNA is called a nucleotide. So if we're thinking of a recipe book and a recipe or a particular protein calls for flour, then one nucleotide is like the F, one is the L, one is the O, one is the U, one is the R. So it's like the most basic building block, the one letter that together makes up the recipe to make you, if that makes sense. So this is a nucleotide. This is one, this is one, this is one, and we'll talk about what makes up a nucleotide. So the first thing in every nucleotide is a phosphate group. So that's a phosphorus with oxygens. That's a phosphate group. And then that is bound to a sugar. So this particular sugar in DNA is called deoxyribose. You may have heard DNA called deoxyribonucleic acid, or DNA. And that comes from the fact that this sugar group is deoxyribose. Okay? Down here I did draw ribose. So that's a different sugar, and you may have heard of RNA or ribonucleic acid. So the main difference between RNA and DNA is that RNA has the sugar ribose instead of deoxyribose. That's where we get RNA and DNA. And the reason it's called that is because ribose has this extra oxygen right here. So without the oxygen, we have deoxyribose. Hopefully that makes sense. And then this deoxyribose is bound to a nitrogenous base. And this is what actually distinguishes the different nucleotides from each other. So every nucleotide has the same sugar and phosphate, but this is what makes them different, and what, say, makes the difference between an F and an L and an R in that recipe. So then the sugar is then bound to the next phosphate group, and so on, and that's what makes the backbone of the DNA. So it's all of the sugar and phosphate bound together that make that backbone, and then they're bound to the nitrogenous bases. And you may know that DNA is a double helix, meaning, like down here, we have two backbones with the nitrogenous bases bound together in the middle. So one strand, this is one strand, would bind together with another strand to make that double helix. So that's kind of the basic of DNA. And now we can talk about the different nitrogenous bases that make up DNA. So in DNA, you have 4 nitrogenous bases. Those are thymine, cytosine, adenine, and guanine. So T, C, A, and G. It's backwards. Those are the essentially letters that make up the DNA, and it's the combination of those that lead to that particular protein that we are trying to make. And C and T, thymine and cytosine, are what are known as pyrimidines, and adenine and guanine are known as purines. The main difference there, these have 2 rings, 2 carbon rings, and the pyrimidines only have 1. And when we are making the double helix, the double strand, the same nucleotides always bind to each other. So in 2 opposite strands, a thymine will always bind to an adenine, and a cytosine will always bind to a guanine. So you can see down here, I drew that like orange always binds with green, and blue always binds with purple. That's just how it is. It does make sense that it's always a pyrimidine and a purine, because these are bigger and these are smaller. And so if you have a double helix, say you had 2 pyrimidines binding and 2 purines binding, the 2 purines binding would be too big. Whereas if you have always a pyrimidine and a purine, you have the same width the whole way down the DNA. And then over here we have uracil, that is the 5th nitrogenous base, and uracil is only present in RNA. So RNA is a different molecule, it's single-stranded, and uracil replaces thymine in RNA. So in RNA we have cytosine, uracil, adenine, and guanine, and no thymine. Don't ask me why, I actually don't know. I could look it up, but I don't know. But that's just how it is, okay? And to replace thymine, the uracil always binds with adenine in RNA. Just something to remember. And I did actually look up like what the purpose of the ribose versus the deoxyribose is, and the reason is that this extra oxygen, the OH, makes RNA much more reactive, much less stable, and that's important because DNA needs to be that stable recipe book. It's in all your cells, it's constant, it has to stay there, so we want it to be really stable and non-reactive, whereas RNA is more of a transient molecule. It is the messenger pigeon that goes and sends things and goes and does transient jobs, so it's better for RNA to be more reactive and less stable. So that's why that is there. The last thing I'll say is that you might see these numbers. So this is essentially numbering the carbon molecules. So each black corner is a carbon molecule, and we number them 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. And the phosphate always attaches to the 5 prime carbon. I like to think 5 phosphate, they sound the same, reason is from the 5' to the 3' direction, and we'll kind of talk about that in the next little section. I always want to say on the next slide, like I'm a teacher with a PowerPoint. But yeah, 5' to 3'. So let's move on to our next page, and this one's kind of similar to what I showed in the last video, but a little bit different. Okay, so we're going to talk about how that DNA makes the proteins. So here we've got our DNA. This is a chromosome, so you may know that your DNA is broken up into 46 chromosomes. Well, 23 unique chromosomes, and then you have 2 of each of those to make 46. And so this is just showing that a chromosome is like all of this coiled DNA wrapped up. And then this is like an expansion of what that looks like. And we've got our A's and T's, our G's and C's. So first we'll talk about what's called DNA replication. And this is just what it sounds like, the replication of the DNA, just making more of it. And this is what happens when your cells need to divide. So for cells to divide, to make new skin cells, heart cells, liver cells, brain cells, whatever, your body has to replicate that DNA, make a new copy of it in order for the cell to divide and for both of the new cells to still have the same amount of DNA. It's got to replicate. And so when that happens, like I showed before, it happens in the 5' to 3' direction, and you get new DNA molecules. And that, so what happens is that the DNA unwinds, and then each strand serves as a template for a new strand. And so the new strand will be complementary to each of the template strands. So wherever there's an A, the new strand's going to have a T. Wherever there's a G, there will be a C, and so on. And the important thing is that DNA replication is what's called semi-conservative. And that means, like I just said, we're not getting an old DNA molecule and a new DNA molecule. We're getting a DNA molecule that has one old strand and one new strand that's complementary, and then the same thing again, an old strand and a new strand. So that's what's shown here with the fact that we've got this navy blue DNA, and then here we've got one navy blue strand and one red strand, one navy blue strand and one red strand, because it's semi-conservative. That just means, yeah, I told you what it means. So that's replication, but we're gonna mainly be talking about how to make proteins. And so to make this into a protein, essentially taking flour and sugar and making it into our cake, we first have to do transcription. And so this is essentially the same as replication. It's just that rather than a new DNA strand, we are making a new RNA strand. So ribonucleic acid, single-stranded. You can see it's not coiled, it's single-stranded. And this is called messenger RNA. So it essentially sends the message of what that recipe says, a copy of the recipe to say, here, this is exactly what you need to make. And so it happens the same way as replication, in that you'll get the complementary nucleotides based on the template strand. The only difference, as I said, is that the T's will be replaced with U's. So we have uracil instead of that thymine. And once again, this happens in the 5' to 3' direction. So transcription makes a messenger RNA molecule from the template strand. Then it does kind of look like a T, but it stands for transfer RNA. And so what happens here is, so like we said, we've got our nucleotides of the RNA. So let's look here. We've got an orange, a brownish, I don't know, this was like maroon when I did it, but it looks brown, and then a purple. So that's A, U, G. A, U, G are our first 3 nucleotides. So the tRNA is going to come in and it's going to recognize what those nucleotides are. And so there is a specific tRNA for each 3 nucleotide combination that can be made. So that is called a codon, the 3 nucleotides that send the message, kind of like each individual word in a recipe is a codon or 3 nucleotides. So this specific tRNA says UAC, and that's what's complementary to AUG, because U binds with A binds with U, C binds with G. So it's complementary. It recognizes that first 3 nucleotides. And whenever we have a UAC tRNA binding to an AUG, it is bound to an amino acid called methionine. Methionine. And that is the start amino acid. So every protein, every mRNA strand starts with an AUG for methionine. That's how the tRNAs know that that's where to start. The translation is an AUG. And this chart down here shows each of the possible codon combinations and what amino acid that codes for. So if you look, first letter A, second letter U, third letter G, you get that methionine. And the same goes for all of the other ones. You look and you see which amino acid. You can see that there are a lot of codons that code for the same amino acid. So all of these code for leucine. That is essentially because if you have 4 nucleotides and you're making combinations of 3 of them, there are actually 64 different combinations that can be made. However, there are only 20 different amino acids that exist, 20 different building blocks of proteins, and so some codons code for the same amino acid. It essentially helps because sometimes if there's a mutation, if one nucleotide is changed, it could still code for the same amino acid, which wouldn't lead to any disease or any impact. So it's kind of evolutionarily good. We'll talk about that a little bit more next. But so there are tRNAs for every possible anticodon. So this is an anticodon based on the codon. tRNAs come in and they bring the amino acid to form the amino acid chain that is coded for by the mRNA. So let's look. We've got our methionine because of our AUG. Our next nucleotides are AAG. Sorry, my hand is shaking. My arm. AAG. So let's look and see if you can figure out where that is. Right, so we've got our A, and then we've got our A, and then we've got our G. So that codes for lysine, and that's why we've got our lysine as the next amino acid. And then next we've got, let's see, AAG, U, 3 U's in a row. So let's find it. U. That codes for phenylalanine. So we've got our phenylalanine. stop. That's a stop codon, which essentially means that we are done coding. So from the AUG to the stop codon, in this case UAG, or UGA, that is the protein coding sequence, and then we are done. So you can see that histidine is the last one because that stop codon doesn't code for an amino acid. We're done. We're ending this protein chain, stopping it and making the protein. And there you can see that there are two other stop codons, but in this case we've got UGA, and that makes our little amino acid chain of 4 amino acids. And then from there it's going to fold up into whatever protein it's supposed to make, and we get our protein, whether that's a liver enzyme or collagen. I don't know. It's going to be a protein. So that's what we get, and it does a particular job in our body. I think that's what I wanted to say. Replication, transcription, translation, protein. That is how we get our final product from our recipe. Okay, so that's the basics. Now we're going to talk about what happens when things go wrong. Okay, so this page is going to be a little bit busy, but bear with me. We're just going to go step by step. Okay, so we've got a 4 codon chain here, or a 3 amino acid chain. What we've got is our start codon, methionine. We've got a proline, a glycine, and then a stop codon. So that is our normal chain. Let's say this makes an important protein for our body. That's the normal train that we're supposed to get, okay? That's our protein. So now we're going to look at what happens when we have mutations in our DNA. So a mutation is just a change from what is normal. A changed nucleotide, an inserted or deleted nucleotide, any change from what is normal. And we're going to look at those different types of mutations. Okay, so in the second row here, AUG, normal. Here we've got CC and then a U instead of an A. U instead of an A. So this is our point mutation. One changed nucleotide, A to U. Well, if we go back and we look at our codon chart, if you look at CCA, so CCA, we have proline. But if you look at our new nucleotide, CCU, we still have proline. So that's what I was talking about, where it codes for the same amino acid regardless. We still have a proline there. So that is what's called a silent mutation, when there's a change in the DNA, but it doesn't change our protein at all. Silent mutation. So there's not going to be any effect of that. Next, here, our one changed nucleotide is going to be this G. So this C is changed to a G. And when we look at the codon chart, that does change the amino acid that is coded for. So instead of our proline in this position, we get an arginine, a new amino acid. Glycine is still the same, stop codon still the same, but we have a different amino acid in the middle. So this is called a missense mutation, when we have a changed amino acid. And totally depending on just depends on what protein, what part of the protein we're talking about, to determine what the effects are going to be. So that's actually a job that I would enjoy doing, is called variant classification. So essentially looking at mutations and trying to determine if they have an effect on the end product, or like, you know, how it functions. But yeah, doesn't always have the biggest effect. Sometimes it has a really big effect. So that's missense. Next, so here we've got our methionine, our proline, normal. Here we are changing our G to a U. We have a U instead of a G. And when we look at our codon chart for UGA, this now makes a stop codon instead of a glycine. So this is going to make our translation stop prematurely, because we have this premature stop codon, and that is called a nonsense mutation. When we get a premature stop codon, most of the times these are bad. Most of the time they lead to a big issue. Here, if we're changing one amino acid, sometimes the protein still folds normally. It still makes a protein that functions more or less like the old one. If we're here and we have a premature stop codon, let's say only a fourth of the way through the protein coding sequence, like here it's at the end, it doesn't change much, but if it's in the middle of the sequence and we're missing half of our protein, it is not going to function the way it's supposed to. If that's our collagen or whatever, we're not going to get a protein at all. The body's just probably going to throw that away because it's so different from what it was supposed to be. So usually these are an issue. A nonsense mutation. Now we are going to look at the last type of mutation that we're going to talk about, and this one is an insertion of a nucleotide. So we still have our AUG, but then in the middle of our CCA, we have inserted a U. So we've inserted one nucleotide there, and what that does is it means that now CUC is our second codon, and AGG is our third, and then AUA is our fourth, and so on. So when we're looking at the codon chart, obviously our methionine is the same, but every other amino acid is changed. So we've got a leucine, an arginine, an isoleucine. Every amino acid is completely changed because of that insertion. So this is what's called a frame shift mutation, because it is shifting our reading frame. So we no longer have the same reading frame, the same codons as before. Whereas like if we change one, the reading frame remains the same. This next amino acid is still the same. We keep it the same, but this shifts everything. So that's a frame shift. It can happen due to a deletion of a nucleotide, or insertion of a nucleotide, or multiple nucleotides. So if you insert 3 nucleotides, you're gonna get one extra amino acid, but it's not actually a frame shift. Because like if we inserted 3 right here, then after that amino acid we would still have CCA. So it wouldn't shift the frame. It only shifts the frame if there's anything other than a multiple of 3. So 2, 4, 5, 7 nucleotides that are inserted or deleted. So yes, this is like we need flour for our recipe. This is like we change change the O in flower, but we still know that we need flour, right? This is like, okay, now we're gonna use almond flour. Slightly different, might still work, might not. This is, we don't even have flour. We don't have flour in the house. And then this is like, I don't know. Like, we just changed every ingredient. Every ingredient is now different from what it was before. So those are our point mutations, different types of mutations. There are lots of other ways that we can get diseases, like issues with the chromosomes, but this is looking at the sequence specifically, so how we can change the sequence itself to get different changes in our DNA. Okay, next we are going to talk about some of the inheritance of these changes, like the mutations we talked about, but how that can actually lead to disease and be inherited. So, the first type of disease we're going to talk about is autosomal recessive. I know I've talked a little bit about this before, but autosomal recessive. So, autosomal is referring to our chromosomes that are not our sex chromosomes. So I said we have 23 unique chromosomes. We have 22 that are autosomes, so everybody has them, males, females, they're exactly the same. And then we have one set of sex chromosomes, that's our X and Y, that are different between males and females. So autosomal recessive, we're talking about mutations on autosomes, which also means that they are going to affect boys and girls the same. No more boys are going to have it than girls, if that makes sense. And I also said that we all have 2 copies of each chromosome to equal 46. We get 1 copy from our mom and 1 copy from our dad. And I also said that chromosomes have genes on them, so they're made up of a bunch of different genes. So here we've got this chromosome with this mystery gene in green. We don't know what gene it is. We'll say that it's the gene causing cystic fibrosis, which is called CFTR. CFTR leads to cystic fibrosis. So we'll say this is the CFTR gene on the chromosome we get from mom and the chromosome we get from dad. And they're both green, meaning we're good to go. We have normal CFTR on both chromosomes, making 2 copies of good CFTR, no cystic fibrosis. Now let's say that we have one of those normal copies and one that's mutated. So maybe that's one changed nucleotide, maybe it's a deletion of nucleotides, maybe it's a deletion of the whole gene. Anything that makes it not work properly is where we get a mutation. So we got one mutation from mom and we got the normal from dad. So this is called being a carrier of the disease. You carry the mutation, but because you still have a normal copy, you don't have the disease because you still have that normal one. Now, in the case of cystic fibrosis and sickle cell anemia and a lot of others, carriers can sometimes have symptoms, sometimes have slight issues because of that mutated gene, but typically we say that they don't have that disease, they don't have a diagnosis of that disease. They might have slight symptoms, but nothing like someone that actually has the disease. So that's a carrier. Now, with autosomal recessive, we have the disease if we inherit 2 copies of that mutation. So one from mom, one from dad, which would mean that parents had to have been carriers. Both parents had to have a copy of that mutation to each pass it on to us. And that is where we get the disease like cystic fibrosis. And something about autosomal recessive conditions is typically these mutations are called loss of function mutations. So what that means is the disease is caused when our protein is not functioning properly. Sometimes mutations can give a protein a new function, make it super active all the time, make it do something it's not supposed to do. Loss of function mutations happen when, say, the gene is deleted or something makes it non-functional. Loss of function. And that's kind of shown by the fact that if we have a mutation but we have a normal copy, this normal one is still doing that function that we need it to do. So having the deleted one isn't the biggest deal because we're still doing that function. We still have CFTR. We'll talk about autosomal dominant conditions next, but it's kind of the other story. But yeah, so loss of function mutations, autosomal recessive disease. So we will talk about this inheritance a little bit. So this is a pedigree. This is essentially a family tree. So squares are males, circles are females. And so these people are married and these people are married. So these are the grandparents. This is their daughter, this is their son, and they are now married. So these are the parents. When these parents have kids, we now have, let's say they had a boy and a girl that both have cystic fibrosis. So they're colored in because they have the disease, and they have a girl and a boy without cystic fibrosis. Well, nobody else in the family has cystic fibrosis. We only see it down here. So what this means, if these two kids both got two mutations, means that both parents are obligate carriers. We know they must both be carriers in order to pass on those mutations to their children. So this is the pattern you'll see when looking at autosomal recessive conditions, is that you'll see children that are born with the condition with no family history of it. Because maybe grandma is a carrier, but if grandpa was not a carrier, none of their kids would have gotten cystic fibrosis, because both parents have to be carriers. So only their kids are carriers, and then this disease only showed itself when two carriers got married and had children. So that's what you'll see, is kind of a condition coming out of the blue. And this just shows a pedigree of inheritance. So essentially, let's say this is mom. So mom has a normal CFTR gene and a mutated one. And dad also has a normal and a mutated one. So now when they have kids, they each have a 50% chance of passing on either gene that they have, either copy. And so these are the four possibilities that can happen. Either both of them pass on their working copy and we get a child who is completely normal. One of them passes on their working copy and one passes on their non-working copy, and we get a carrier. So somebody that doesn't have the disease but carries a mutation. Or they can both pass on the mutation and we get somebody with the disease. So that is what has happened here. These kids might be carriers, they might not be, we don't know. So that is what we get. So 25% chance of normal, 50% chance of being a carrier, 25% chance of disease. obviously this is 50%, but anything can happen. You know, it's not always going to be the exact percentages, but with each child they had, there was a 25% chance of getting that. Yeah, the other thing I'll talk about is that the carrier rates for a lot of these diseases are relatively common. So in the Caucasian population, there's about a 1 in 25 rate of carriers for cystic fibrosis. 1 in 25 Caucasians is a carrier of cystic fibrosis. Fairly common. What's not so common is 2 carriers getting together and then having a child with cystic fibrosis. So what those chances are is there's a 1 in 25 chance of you being a carrier. Let's say you're Caucasian. 1 in 25 chance. The chance of you also marrying a carrier is 1 in 25, or having kids with a carrier, also 1 in 25. So the chance that 2 Caucasian people, random Caucasian people, are carriers is 1 in 25 times 1 in 25. Then, if they both are carriers, the chance they'll have a child with cystic fibrosis is 25%. So 1 in 25 times 1 in 25 times 1 in 4 gives you a 1 in 2,500 chance, or 0.04% chance, that any 2 random Caucasians are going to have a child with cystic fibrosis. So 1 in 25 carrier rate, 1 in 2,500 chance of cystic fibrosis. And this, I always like to say, is why incest is bad. Obviously, there's the moral stuff, the ickiness, right? Like that we think about, but genetically, the reason that incest is bad is because if you marry someone that is related to you, there's a really high chance that they are going to be a carrier of the same things that you're a carrier of. So if you go meet a random person on the street, you're probably both carriers of something. There's a really low chance that you're both carriers of the same thing. Whereas, let's say these grandparents have kids and they marry their cousin, well, there's a really high chance that both of them and their cousin are carriers and got it from the same person. If you share a grandparent with somebody, you probably have some of the exact same genes as them. And so if you have kids with them, there's a really high chance you're gonna see a recessive condition in the kids. So that is why marrying relatives leads to genetic conditions, is because you have some of the same mutations, some of the same genes. Okay, so that's autosomal recessive. Now we'll briefly talk about autosomal dominant. We already kind of went over a lot of it, so less to talk about. But with autosomal dominant conditions, we only need 1 copy of that mutation to get the disease. So here we've got our normal to normal copies, no disease. Here we have a normal copy from one parent, and we have a mutated copy, and that gives us the disease. So this is more of what we call gain of function mutations. So it kind of makes sense when you think about it. Loss of function, we would need to lose both copies of that gene to get the disease, because if we lose one, we still have the other. Whereas if we have a gain of function mutation, something where this gene is now overactive or it's doing something it shouldn't be, well, it doesn't matter if we have a normal gene there. That gene is going to cause some trouble because it's doing something it shouldn't. you can't carry the disease and not actually have it, because anyone with a mutation has the disease. So we see, we'll talk about the fact that grandparents don't have it in a sec, but we see dad has this mystery autosomal dominant condition. We'll call it achondroplasia or dwarfism. Achondroplasia is the word for the most common type of dwarfism. Let's say he has that. He has kids. A girl and a boy both get it, and a girl and a boy do not get it. So 50% of his kids got that from him. Over here we can see the pedigree. So mom has 2 normal copies. That's the only thing she can pass on, is a normal copy. Dad has 1 normal and 1 abnormal. And so we get 50% disease, 50% normal, which is what we see here for these 2 parents. Now, something that happens with a lot of autosomal dominant conditions, obviously it can be inherited from a parent, but these mutations can sometimes be de novo. So they can happen randomly, they can happen without being inherited from a parent. And that kind of happens because all we need is 1 change in the gene to cause the disease. So sometimes that can happen when DNA is replicating, when an embryo is being formed. We can get a random mutation, and it's not necessarily passed on from the parent. So it's a de novo mutation. And that's what we're seeing here. Normal parents having a child with the condition is a de novo mutation. And then from there, dad can now pass it on to his children because he has it, he carries it. So with achondroplasia specifically, the de novo rate is 80%. 80% of people with this kind of dwarfism have parents that do not have dwarfism. They're perfectly fine. So, yeah, 80% de novo rate, but then when they have kids, there's a 50% chance that their kids would have it. Yeah, I think that's everything that I wanted to talk about there. I will say, if both parents have achondroplasia, so they both have a mutation and a normal copy, now it's true, the pedigree would be like for a recessive condition. So there would be a 25% chance that both of them would pass on the mutation to their child, and that the child would get 2 mutations. And specifically for achondroplasia, that's lethal. So we have that gain of function mutation, but we still have the normal copy. We get achondroplasia. If we had 2 gain of function mutations, it's lethal. It would end in a miscarriage if a child had that. So that's one thing with 2 parents with achondroplasia. There would actually be a 25% chance that the baby would not survive, 50% that they would have achondroplasia, and then 50% normal, which for living children comes out to a 2/3 chance of achondroplasia and a 1/3 chance of normal. Sorry, I keep saying normal. I shouldn't say that because I don't want to call people with conditions abnormal. Like, not abnormal, it's just that's what we say is like a normal gene, you know. I'm not saying the person is normal or abnormal, but the gene is the way that it normally is, or is not the way it normally is. Yeah. Sorry if I ever say like language things that are weird. I don't know. Okay. The next type of mutation that we are going to talk about is somatic mutations. So far we have mostly been talking about mutations that are inherited, that are in the germline. So if a mutation is inherited, it is in your mutases is when it's actually passed through your sperm and egg. However, sometimes mutations are not in the germline, they're somewhere else in your body. So that's what we'll talk about. So here, this is a germline mutation. So let's say sperm has the normal copy, egg has the mutation. When those combine and form an embryo, now every single cell of the embryo has that mutation, that red dot. So when we get the baby or the person, they have that mutation in every single cell of their body, including their sperm and egg. So if they have a child, then they would be passing on that mutation, or the possibility of passing on that mutation. So this is like our cystic fibrosis or our achondroplasia. It's in every cell of the body. Now here, let's say sperm and egg do not have the mutation. They are perfectly normal. We form an embryo, and now the embryo is normal. Sometimes you could get a mutation here in one cell of the embryo. Sometimes it could be after. It could be during your lifetime, or it could be at some point before birth that you get this mutation. But the main thing is that it's not in every cell of the body. So now we get a person who most of their cells don't have the mutation, but some of them do. So this is kind of cartoony, but we'll say that some of the cells on their head or whatever have this mutation in it, but it's not in their germ cells. So it cannot be passed on, but it can cause disease in them. There are examples of this for like a disease that you're born with. There's a disease called, what was it? McCune-Albright syndrome, I think, is like a skin condition, and it's always a somatic mutation. So it's not something that's passed on from parents, and it's not something that affected people can pass on. It's just a mutation that happens like in embryo formation, and that person has the disease, but they don't pass it on. But another main example of somatic mutations to discuss is cancer. So throughout your lifetime, you acquire mutations in your DNA that can cause issues. And it can be things like carcinogens that lead to those mutations. So they damage your DNA and cause changes in your DNA, and that can lead to cancer cells, which are essentially cells that have damaged DNA. And that can cause cancer due to those somatic mutations. However, you are not passing those mutations on to your children. If you lay out in the sun every day of your life, that UV light is gonna damage your DNA, and you're gonna be at a high risk of skin cancer. However, you're not going to pass on that risk of skin cancer to your children because it's somatic. It's not in your germline. It's not in your eggs and sperm, so you can't pass it on. So these somatic mutations can happen due to errors in your DNA replication that happen normally, errors in the repair of DNA, because normally your body has repair mechanisms that repair those mutations in DNA. Sometimes they don't work, and it can happen due to carcinogens, environment, any type of mutagen, or something that causes mutations in your DNA. So that is the difference between something germline and something somatic, which essentially, somatic means like body, so your body cells rather than your germ cells, which are the ones that make a baby. And that's also why I say that all cancer is genetic. It's all due to mutations in your DNA. Not all cancer nucleotide being deleted or added, a nucleotide being changed, mutagens causing mutations in our DNA. This we're going to be talking about extra or missing genetic material on the order of the chromosome. So here we've got mom. She has her normal 46 chromosomes. So I only drew 2 pairs, so the purple pair and the green pair. Just imagine she's got 23 of them. And XX because she's a female. And then same with male, he's got, or with dad, he's got both pairs of all of his chromosomes as well. So 46, 46. So now when they are having kids, we go through what's called meiosis, which is where it makes the egg and the sperm that is going to combine with the other gamete. So if you're a female, it makes the egg that combines with the male's sperm. And each of those eggs and sperm is supposed to have one of each chromosome. So 23 chromosomes, one of each. And when that combines with the other gamete, the other 23, we get a pair of each one. So 2 of each of those chromosomes. Normally, that's what's supposed to happen. Well, here we see that when the female underwent meiosis and made 2 eggs, I'm simplifying this, there's another step involved, but I didn't want to draw it all. But when she made her eggs, each one got a normal purple chromosome. So each one got one like it's supposed to. But the green ones didn't separate properly. So they both went to this cell, and none of them went to this cell. The dad, everything went normally. Both sperm have a purple and a green. Well, now, let's say this egg combines with a sperm, and we get this zygote. This zygote has 2 purples, like normal, but it only has 1 green from dad. So that is known as a monosomy, meaning only 1 chromosome. We're supposed to have 2, but we've got a monosomy. So we are going to call the green one chromosome 21. 21 of 23. And this baby is missing a 21, and that is lethal. You can't be born missing a 21st chromosome. It's too severe of a problem. But here, if this egg combines with a sperm, we get our same 2 purples, but we get 3 of that 21st chromosome. 2 from mom, 1 from dad, 3 total. So this is an extra 21st chromosome, which is known as a trisomy. So 3 chromosomes. Trisomy 21, also known as Down syndrome. So this is how we get Down syndrome if we have 3 of that 21st chromosome. And so, having extra is better than missing. Missing typically is not survivable if you're missing a whole chromosome. Having an extra one can be in the case of Down syndrome. Most other chromosomes, having an extra one is more severe, but the 21st chromosome is the smallest, so having an extra, it leads to Down syndrome rather than being lethal. And so you can have issues on the order of the whole chromosome, or you can be missing or have an extra of a little part of it. Maybe I'll talk about that in another video, but there are a lot of deletions and duplications of small parts of chromosomes that can happen, that have a wide range of effects. I've kind of talked about some of those before. Okay, so that's all of the types of mutations that we are going to talk about. The last page that I'm going to show you is kind of going back to that mental health and everything like that. And that is on multifactorial disease. diseases where a mutation causes a disease. In cystic fibrosis, deleting that CFTR causes cystic fibrosis. Cause and effect, easy peasy. One causes the other. The vast majority of diseases, mental health, heart disease, diabetes, Alzheimer's, everything, are multifactorial. Meaning, yes, there are genetic factors. There are genetic factors that have to do with everything. But there are also environmental factors. So it's not that one gene causes one disease, that there is a gene for depression and you get depression. It means that you have so many genes and there can be so many differences in those genes. We all have differences in our genes, and maybe your genes have a few more predisposing factors that lead you to have a higher chance of diabetes, for example. So there are genetic factors, but it has to be combined with environment to cause the disease. So multifactorial. And this is it's kind of like the beads in a jar theory or whatever. Essentially, you have to have enough factors to overfill this jar to get the disease. If we're not overfilling the jar, we don't get the disease. So here, we've got our genetic factors and our environmental factors. So we've got an average genetic risk. Each of these beads are like something that might put you at risk of, we'll say, diabetes or heart disease. Average genetic risk and average environmental risk. We'll say you lead a pretty healthy lifestyle. You don't get the disease. You're not overflowing. You're good. Here, we have the exact same genetic risk. We'll say average genetic risk. But we have a ton of environmental risk factors. So let's say this person has the same genetic risk as this person, but this person has smoked a pack a day their whole life. They drink a lot every single day. They don't exercise. They're overweight. Now they have environmental factors that are overfilling their jar, and it leads to diabetes. So the genetics are part of it. The genetics put them at a little bit higher of a risk, but it was the environment that sent them over the top. Now here, we have the same environmental risk as here. This person leads a relatively healthy lifestyle, but they were born with a lot of genetic risk. Let's say many of their family members had diabetes. They're born with quite a few genetic factors that predispose to diabetes. Maybe doesn't necessarily mean they're gonna get it because their jar is not full of genetic factors, but just eating slightly unhealthy or not quite exercising enough is enough to push them over the edge. So that kind of explains why some people can be at a higher predisposition and it takes less to push them over that edge to get that disease, whether it's mental health or diabetes or heart disease. Other people are maybe born with not too much genetic risk, but their environment is what pushes them over the edge. So multifactorial basically shows, how like some people get diabetes and it was a lot of genetics. Like they live a pretty healthy life. They were just predisposed from the get-go. And other people, it's because of their lifestyle that they get it. No one in their family got it. They have average genetic risk, but it's their lifestyle that caused that. So that's kind of touching on the mental health and things like that, is that it's a combination of a lot of factors. It's not one thing causing it. It's genetic, but it's also other things. So yeah, us to the end of this video. I hope you guys really enjoyed it. I had fun. I've been talking for over an hour now, so I'm gonna go rest, but I hope I said everything that I wanted to say, and I hope that you guys enjoyed it. Please let me know if you have any questions, anything like that. If you go to my school, let me know. I don't know. Yeah, so thank you everybody for watching, and I will see you in my next video. So sleep well, everybody. Sleep well.